For the inflammatory response, we're going to use a Torrey Pines and CCA analogy. In this analogy, Meglaine, Anne, and Megan represent mast cells and macrophages by the site of injury. The pathogen, Isabel's ex-boyfriend, enters the body from Torrey Pines. This causes the mast cells and macrophages, Meglaine, Anne, and Megan, to send out the histamines and other signaling molecules in the form of snapshots. These snapshots cause CCA, the capillary, to open the doors and increase student flow. Isabel, a type of white blood cell called a neutrophil, leaves CCA and is attracted to her ex-boyfriend, the pathogen. She then proceeds to kill him.
An activated T cell sends out cytokines, which initiate either a humoral or cell-mediated response. The cell-mediated response involves the activation of cytotoxic T cells, and the humoral response involves antibody-creating B cells. For the cell-mediated response, an infected cell displays a pathogen's antigen fragments with the use of class I MHC molecules. A cytotoxic T cell recognizes the antigen fragments displayed by the class I MHC molecules on the infected cell and binds with the help of CD8 accessory proteins. The T cell then releases perforin and granzyme molecules. Granzymes break down proteins with the infected cell, and perforin molecules cause pores to form. Fragmentation of the nucleus and other components of the infected cell leads to apoptosis and eventual cell death. For the humoral response, a B cell differentiates into memory B cells or plasma cells. Memory B cells will remember the antigen of the pathogen for future production of antibodies. Plasma cells will produce thousands of the same antibodies that are released into the body for neutralization or destruction of this specific pathogen.
Antibodies produced by these plasma cells participate in two main processes: neutralization and opsonization. In neutralization, antibodies bind to the pathogen's antigens and coat its surface, hindering the pathogen's ability to bind to a healthy cell's surface receptors and infect it. In opsonization, antibodies bound to the pathogen market for destruction by phagocytes. In this case, the unbound branches of the antibodies are recognized by receptors on the surface of the macrophage, allowing it to engulf and destroy the pathogen.